Hey everyone, welcome to today's OBSSR Directors Webinar. I'm Jane Simone. I'm the Director of the Office of Behavioral and Social Sciences Research here at the National Institutes of Health. And before I introduce today's speaker, I have a few housekeeping items. First of all, we will be recording this webinar, and then it will be posted on OBSSR's website in about a month. And today's presentation will be followed by a Q&A session. To ask a question or comment, please use the Q&A button at the bottom of your Zoom screen, and feel free to ask questions at any time during the webinar. Please note that the chat feature is disabled, and all attendees will be muted during the webinar. Following the presentation, OBSSR's Dr. Kristen Brethel Horowitz will facilitate the Q&A and ask your questions to the presenter. Now, I'm very pleased to tell you a bit about our speaker, Dr. Michael Platt. Dr. Platt is a professor and director of the Wharton Neuroscience Initiative at the University of Pennsylvania. He is author of the book, The Leader's Brain, and has published more than 300 articles. He previously served as director of the Duke Institute for Brain Sciences and the Center for Cognitive Neuroscience at Duke, and was the founding co-director of the Duke Center for Neuroeconomic Studies. His work has been featured in outlets such as the New York Times, Washington Post, Wall Street Journal, Newsweek, The Guardian, National Geographic, as well as on ABC's Good Morning America, NPR, CBS, BBC, MTV, and HBO's Vice. He currently serves on the advisory boards of several companies, as well as the Yangtan Autism Centers at the Massachusetts Institute of Technology and Harvard. He previously served on the World Economic Forum's Global Future Council on Brain Science, was president of the Society for Neuroeconomics, consulted on Darren Aronofsky's film, The Fountain, and co-founded Cogware, a company focused on brain health and performance. Please join me in welcoming Dr. Platt.
All right, well, thank you. It's a real pleasure to be here. And I'm really excited to share some of our work. I'm going to keep this pretty high level because I have a lot of slides and we want to make sure that we get to tell the whole story. So I've been studying non-human primate and human primate social behavior since I was actually in college. And so it's kind of a long journey to tell you about, but really what's the point? What's the purpose? And here's the message. We are wired to connect. I mean, this is the secret of our success as a species, I think, beyond any of our other capacities. And we know from really robust epidemiological evidence that people who have more social support, so better friends, more friends, live longer and healthier, happier lives, and they also make more money because having good social skills, social emotional skills is really good in business as well. And we know, unfortunately, painfully from studies coming in over and over and over the last decade, and in particular since the pandemic, that the lack of social connection is not just bad for our mental health, but bad for our physical health overall. So we know the work of Paul Lundstedt, for example, has shown that not having social support, being lonely, is worse for your health than, say, smoking 15 cigarettes a day. So it's this, I think, these data really demonstrate how core it is to us, our ability to function, to survive and thrive and perform, that our social ties are. This is not uniquely human, although it, I think, finds its grandest expression in us. But social factors are a major determinant of life, of health, in a bunch of non-human animals shown here in this schematic, that basically having more social capital in the form of more connections, more friends, more allies, as well as social status, and those two often go together, is a major determinant of outcomes for a variety of animals. So, for example, monkeys and antelope and whales and horses and marmots, you get the picture. You have more friends, you have more status, you basically live a longer life, and in many of these species, you also tend to make more babies. So it's an asset that helps one to thrive. Now, how to understand the underlying mechanisms? I mean, that's a big challenge, and there are a variety of approaches that have been taken. Our approach, where part of our approach takes place here, which is a long-running field site on Cayo Santiago off the coast of Puerto Rico, founded in 1938 with a population of roughly 400 rhesus macaques from across South Asia. At any one time, there are 1,500 to 2,000 monkeys living without any major human intervention, certainly no veterinary support. So they live their lives, they do what they do. And we focus on rhesus macaques because they are closely related to humans, share the preponderance of our DNA. They are wired to connect basically in the same way that we are, as I will show you. So they have the same basic circuits in their heads that manage their moment-to-moment interactions and long-term relations that we do. They depend on each other for survival. And just like humans, monkeys who have more social support, who have more friends, and who have more status actually live longer, and they make more babies. So that you can think of that as kind of the equivalent of making more money. So CAIO is a really special place because we basically have people, boots on the ground, who follow monkeys around all day long and record everything that they do. So we have exquisite, detailed phenotypic, behavioral information, demographic information, life history information, going all the way back to the founding of the population in 1938. And then basically once a year, we conduct a big monkey rodeo where we trap hundreds of monkeys. This has been going on forever, take biological samples. So that begins to give us the capacity, for example, to relate underlying variation in genes or other physiological markers to behavior and to long-term outcomes. And as you'll see, beginning in 2016, we began a program of population reduction and management in collaboration with the NIH and University of Puerto Rico, because we needed to reduce the size of the population. And that was an unparalleled opportunity to get access to other kinds of tissues, as you'll see. So the work that I'm describing in the first half of this talk is not solely my own work. So we have a large group of collaborators that go back working together since beginning in 2006. You can see some of them up there. We got our first NIH support for the work that I'll describe, one of the ERA grants in 2009. We basically had uninterrupted financial support from the NIH and NIMH, Aging, the Brain Initiative, and the NSF as well. Over the course of this time, we started out with really just a couple of people, and now there are over 100 people working with us on this project. And we demonstrated along the way that, again, just like humans, these rhesus macaques depend on each other. Rhesus macaques in this population who have more friends, more allies, so they have stronger social connections, live longer, and they have more babies too. So it's a protective factor for them. Now, in 2017, Puerto Rico was hit by Hurricane Maria, which is the most intense natural disaster to ever befall that island. 3,000 people lost their lives at least, and the hurricane itself unleashed a sort of tidal wave of mental health issues, including anxiety, depression, and suicide. That hurricane hit. So this is sort of the aftermath of that hurricane, and there's a long story to how we continue to support our research staff down there, as well as the monkeys. But that hurricane hit CAIO first, and so it was packing 170-mile-an-hour winds. It was really devastating. We were really concerned that all the monkeys would sort of. I'm going to shut off that audio, but you can see that it was like a bomb went off on CAIO. So all the research infrastructure, all the support infrastructure was destroyed. Basically, all the trees were destroyed. It took a long time to get things kind of back up and running, but our field assistants were, once we determined they were okay, they were back on the ground and continuing to observe monkeys as they had in the past within just a few days. Miraculously, basically, very few monkeys died as a result of the storm. They were, however, forced into a really difficult situation, eating seaweed, drinking seawater until we could get the fresh water collecting cisterns back functioning. And then basically we've been able to chart what happened with these animals in a way that you could not replicate, I don't believe, very easily in the laboratory. So imagine what we've got here is incredibly detailed behavioral, demographic, life history data, and a lot of physiological data, including genetics, on these animals before the hurricane. And then we have the hurricane as a major insult, right? Acute insult with lasting trauma, because as you can see here, one of the impacts of the hurricane was to to basically kill all the vegetation on the island. And as a result of that, Kaya is about 8 degrees Fahrenheit hotter, which is right at, in the afternoon, is right at the borderline of being impossible to deal with physiologically. These monkeys don't sweat. So their only way of dealing with the intense heat and sun is to try to find shade, which is now a very precious and scarce resource. This hurricane and its aftermath had a big impact on the bodies of these animals, beginning at the molecular level. So this is a plot showing the expression of genes in blood that are either turned up or turned down by the experience of going through the hurricane, and they also are turned up or turned down as one ages. So what you can think of here is that the hurricane caused accelerated aging of these monkeys, largely by increasing inflammation and reducing the factors that help to keep DNA intact, to keep it repaired, et cetera. So it's bad at the molecular level. And essentially, this is what I said before, the monkeys on average, just going through 1 year of that hurricane and its aftermath, it's like aging 2 years for these monkeys. For us, that's about 8 years. I certainly wouldn't want to experience that. But you can also see that there's a lot of variation. So some monkeys effectively aged much more rapidly biologically, and some monkeys did not. They were protected from that. And that, of course, is the object of interest for a lot of our work. We see that that increase in inflammation manifesting in a number of ways. We saw increases in the size of the spleen and the liver, which I'm not showing here. Here is indicators of kind of early onset arthritis, if you will. So the blue curve is hip flexion or hip extension, sorry, in monkeys as a function of age. You can see that it's high when they're young and kind of goes down with plateaus while they're adults. And then in old age, it drops off rapidly. So that's like what I'm experiencing right now with my knees. The red curve is a group of monkeys sampled 1 year after the hurricane. You can see that, boy, they're really doing worse. Their hip extension is terrible. And 2 years after the hurricane, even more. So going through that hurricane for these monkeys is like, for a young monkey, becomes effectively like a very old monkey in terms of their mobility. And as I alluded to earlier, in 2016, as a part of our program of population management, we began to remove whole social groups. So we kept them intact so as not to disrupt social relationships within any one group. We remove a whole group after first spending a couple of months intensively observing them, essentially behaviorally phenotyping them, remove them, euthanize them all humanely, acquire their brains, acquire all their internal organs. These are monkeys going from young age to very old, so it's across the entire life course. So it's a really unparalleled resource. So we began that in 2016, 1 year before the hurricane. Hurricane happened in 2017, and we've continued to do so ever since. And so I'm going to show you some data based on our neurobiological data. Half of those brains, one hemisphere in each of those brains is fixed in formalin, allowing us to do imaging and histology. The other hemisphere is frozen in thick slabs from which we've been doing both bulk and single cell RNA-seq and ATAC-seq, so looking at gene expression and the regulation of gene expression. And just a kind of a teaser here, one thing that we found in terms of gray matter volume, for example, is something that has been observed before in other studies of the impact of stress on the brain. So the hippocampus, for example, declines significantly in successive years after the hurricane. Okay, so that's consistent with what's been observed in other studies, both in the laboratory and outside the laboratory. So basically, this living through the hurricane and the aftermath of the degraded environment is affecting their molecular biology, their cellular biology, their bodies, and their brains. So what is some monkey to do under these conditions? Well, the monkeys do what people do in response to natural, at least initially in response to natural disasters or even other disasters, terrorist attacks, et cetera. And what we found is that the monkeys actually bonded together. So rather than fight, with each other over every little scrap, instead what they did was to become friendlier, less aggressive, and monkeys actively reached out and made new friends. So what I'm showing you here are social networks based on grooming. Grooming is like for us going out and getting a cup of coffee or a beer with your friend. It's an active way of building and maintaining a social relationship. And monkeys who groom together are more likely to help each other in a fight, for example, later on. So in A and C, we see for two groups a map of their social networks based on grooming, and you can see that there are some monkeys who have friends, and a lot of monkeys don't really invest much in friendships with anyone. But after the hurricane, this is just 1 year post-hurricane, on the right, you can see that is massive increase in the density of this social network. Basically, all the monkeys now found it much more valuable as an investment to build and maintain ties with other monkeys. Okay, so that's really interesting. What does that mean? Well, we've been able to check on that population and how the monkey's behavior has changed or persisted, and the impacts ultimately on survival. And what we found, now this was a paper we published in Science last year, is that the monkeys didn't continue to groom as actively as they did, because essentially it seems like they worked really hard in that first year to build relationships, and then they were able to utilize those relationships. So we see that monkeys spend a lot more time basically sitting close together after the hurricane, and that's sustained all the way up for 5 years after the hurricane, and their levels of aggression are much lower. This is really surprising, this level of flexibility in these animals. If you've ever worked with a rhesus macaque, you know that they're very despotic. They can be very aggressive. Males have these huge canines. They fight and wound each other a lot. Instead, what we're seeing is that they're becoming a kinder, friendlier animal over time. What does that do for them? Well, what it does is it helps them survive. So what we see here are survivorship curves comparing three terciles of monkeys in terms of the blue curve is those monkeys who have the biggest, densest proximity network. So essentially they have more friends, if you will. And the red curve is the ones who are in the lowest tercile. And you can see there's basically a 50% increase or higher likelihood of surviving during that time period for monkeys who essentially have more friends, who have more partners, who are the least aggressive and the friendliest. And that's super interesting. We're wondering, well, why might that be so? And we think essentially what's going on is monkeys have essentially built these relationships, and they use them to access this scarce resource, which is shade. So you can see monkeys here sitting in the shade of this lone tall tree because it is so much hotter. And the heat is concentrated in the afternoon. In the morning, basically, it's the same temperature in what limited shade is still there, as well as in exposed areas. But in the afternoon, you basically don't want to be out in the sun. So and there's more data in the paper if you want to look at it. This is an active process. It's not, monkeys aren't just like billiard balls who end up kind of, hemmed in within the confines of shade, but are actively kind of working on developing relationships so that they have somebody to sit next to when they need to on the shade. So even in the morning, when they're out in exposed areas, they tend to be closer together to those monkeys that they're going to spend time with in the shade later in the afternoon. Now, the beauty of this is that, of course, as I mentioned before, we have access to biological data, including brain data from these monkeys, comparing before the hurricane to after the hurricane. And what's really, I think, very provocative is when we look at those brains, this is on average here, we see that there are a number, I told you that the hippocampus and some other areas that are stress responsive are shrink after the hurricane, but we also see that there are a number of areas here, part of the major parts of the limbic system, that are actually larger, and they get successively larger in cohorts that have been sampled after the hurricane. So like the amygdala, the superior temporal sulcus, the anterior cingulate cortex. And what's interesting about these areas is that they have all been implicated both in human and non-human primates in social information processing. So whether looking at here in the top row, kind of a much more expanded view of areas that are active in monkeys while they're watching monkeys interact with each other, or here on the bottom, kind of intrinsic signal functional connectivity, essentially, patterns in monkeys paralleling those in humans that suggest that these areas are actually part of the social brain network, which we know is is crucial for managing our moment-to-moment relationships with other individuals, as well as our moment-to-moment interactions, as well as our long-term relationships with others. We know from the work of our colleagues and collaborators, Jerome Soleil et al. From about a decade ago, that these areas are responsive in terms of their size to the size of the social network that one has. So if you basically take monkeys who are living alone and put them into groups of different sizes, and then compare the brains after a few months of living like that to before, you see that there are a number of areas that expand in size, and they're the same ones that we see in our monkeys as well. And in fact, before the hurricane, we had demonstrated that there is a relationship between the size of some of these key areas, like the mid-superior temporal sulcus, MSTS, thought to possibly be homologous with the human temporoparietal junction, as well as the insula and portions of the amygdala scale with how many friends you've got. Okay, and notably, this only begins to emerge post-adolescence as monkeys begin to develop their social networks. So it seems to be in response to use, if you will. The big question then is like, well, what do these areas do? What are the computations they perform, and how are those computations enabling the monkeys to respond with behavioral flexibility to an altered and devastated environment by expanding their investments in social relationships? We study these things. We've been studying these things in the lab neurophysiologically for over 20 years, again with uninterrupted NIH support. And what we do, and this is kind of emblematic of it, we teach monkeys to play games like the one on the left here. It's actually, we have humans playing the exact same games. These are monkeys and humans playing a version of the game of chicken. So it's a little bit complicated, but you've got two individuals facing off against each other. They're controlling those circles, which are cars, if you will. They can see tokens that are that can be converted into reward. So for monkeys, that's juice. For humans, that's money. So there's a temptation to go forward to get as much juice or money as possible, rather than deviate and avoid crashing, which is the thing you don't want to have happen. But in our version of this task, this is just emblematic again. This white bar is a cooperation bar. So they can cooperate if they can coordinate their behavior to unlock some bonus rewards. They generally do that, and they do so when it makes the most sense economically, and also social status plays a factor. So monkeys who are lower ranking tend to cooperate more. Monkeys who are higher ranking cooperate a bit less. But their choices depend on a lot of different factors. So we tried to, basically the beauty of this kind of approach is that we can submit it to formal mathematical modeling, game theory here, implemented also with reinforcement learning. And basically you can't explain the behavior, the choices of monkeys or people with anything kind of simple model, just based on reinforcement learning. Did I get a reward or not? How much was it? Or how much the other monkey got? But instead, you have to include a lot of information with regard to kind of the behavioral tendencies of the other individual, whether they're deviating from those tendencies, how cooperative or strategic are they? And you can see here that you've got to build a really sophisticated model that begins to border on, kind of social reasoning, strategic social reasoning of the sort that we think of when we consider things like theory of mind. Not saying that's necessarily what they're doing. In order to explain their behavior. And the beauty, again, of this approach is we can then look for these various pieces of information in these boxes in the brain. And that's what we did in a study we published a couple of years ago, comparing two parts of the social brain network, the anterior cingulate cortex and the mid-superior temporal sulcus, both of which, as I showed you before, expanded after the hurricane. And what you can see is that neurons in these areas are multiplexing or carrying information about a number of these different factors that we know are combined together to actually drive the monkey or the human's decision. And I'll just point out that in the mid-superior temporal sulcus, again, thought to be homologous to the human temporoparietal junction, or TPJ, involved in theory of mind in humans, that neurons are particularly sensitive in the mid-STS to rewards that are achieved through cooperation. So it seems to be kind of consistent with what we see in humans in terms of strategic reasoning about whether or not to cooperate We're studying now the impact on competition as well. Now, as I think amazing as that is, and it reveals a lot of really interesting complexity in the information processing, even within the monkey social brain network. The behavior of monkeys in the wild is still much more complicated, much more sophisticated. They live in these multidimensional societies where they have friends, they have foes, they have allies, they have kin, and kinship is really important, and of course, social status. And a wealth of ethological data going back to the 1950s indicates that these monkeys seem to know about the relationships amongst other monkeys as well, and so that there's a richness in their behavior, and which these kinds of games that we, no matter how complex they are, that we implement in the laboratory, they never really replicate exactly what the monkeys do in real life. And so finally, in the last few years, again with support from NIH, we've been able to do the DREAM study that I've been planning for the last 30 years, where we, working with a couple of companies, we developed wireless recording technologies, which allowed us to implant Utah arrays in two areas of the cortex, which are one area, the ventrolateral prefrontal cortex, already has been implicated in various kinds of social information processing in prior studies. An area TEO adjacent to the mid-superior temporal sulcus, part of higher order visual cortex, and we kind of thought of it as a bit of a control area. We know it's selective for signaling information about objects and faces and things like that. We can talk about the technology later if you're interested. Same exact technology is used in humans. These Utah arrays have been FDA approved for use in brain-machine, brain-computer interfaces since 2003, I believe. So here's our study, this initial study. We basically let monkeys do what monkeys do. So we had a human observer, we had cameras all around the enclosure, and the first study, which we published in Nature last year, looked at recordings from these two areas in male subjects who either were with their female partner or not. And then we could also vary the presence and identity of a neighbor to change the social context. And monkeys were free to do whatever monkeys do. What we found is that monkeys in the laboratory engaged in a ton of different kinds of behaviors. So about 87% of the repertoire that we observe in the wild, for example, on Cayo Santiago. This is a sort of example of an ethogram of the different behaviors that the monkeys engaged in different contexts, depending on whether they were paired or not paired, whether there was a female observer next to them or a male observer, et cetera. And this is just basically showing that the monkeys are replicating or engaging in most of the behavioral repertoire. I said 87%, 84% what they engage in real life. And you can see that when we look at transition probabilities between the different behaviors, that they kind of cluster into two main groups. One that is much more agonistic, so essentially fighting and threatening, and another set that is much more affiliative, related to grooming and maintaining and building these social relationships with each other. And you can see here that these are the monkeys basically engaging in grooming behavior. The monkey on the right, you can see the recording apparatus on the back of his head, and you can see that neural activity, both a single unit on the top on the right, as well as the population recorded at that particular time, shows strong modulation during behavior. This study is not like other traditional task-based studies where we would typically, you could time lock to the onset of a fixation point, and then the onset of, say, those cars, with the dots indicating rewards, as I showed you before, and then the choice, and then the reward delivery. Here, it's just behavior. And so what we did was to time lock our analyses to the onsets and offsets of these very common behaviors, like grooming. And what we found is, and this is, I think, pretty interesting, is that behavioral states, so we're looking here at neural activity averaged in 500 millisecond bins. It didn't really matter what bin size we used, that there was systematic modulation, graded modulation across the population as you go from kind of more affiliative and relaxed behaviors, if you will, like grooming, getting groomed, et cetera, even foraging, and much more increases in activity when monkeys are moving about a lot and engaging in threats and even outright aggression. And this raises, it's an interesting possibility that perhaps, oh, this is just arousal or attention. I think there's a lot more going on, but we can see that there is there's sort of major modulations and activity within both of these areas equivalently as a function of what the monkeys are doing. It turns out that under these conditions, when monkeys, we've kind of taken the blinders off and they can engage in so many different behaviors, that the average neuron is actually significantly modulated by about 7 different behaviors. So there's much, much greater than you would expect under conditions of sparse coding, for example, like a grandmother cell that only encodes one single thing. So we see that these neurons are multiplexing, if you will, information about multiple different behavioral states, as well as context, as I'll show you in a moment. Now, we were interested in whether, as a population, there may be something more to the representation of all of these behavioral states than just that sort of graded modulation as a function of kind of behaviors going from more relaxed to more energetic, if you will. And so we subjected population activity to the dimensionality reduction analysis using UMAP. And you can see here that in a single example sessions, both for prefrontal on the left and visual cortex, TEO on the right, that in fact the patterns of activity segregate out into separate clusters depending on what the monkeys are doing. So that's interesting. We can decode at a very high degree of fidelity the behavioral state of the animal. But you'll also note that there's this interesting weird pattern going on, which is that there's 2 clouds, for example, in dark blue for monkey getting groomed, and here on the right, 2 clouds for a monkey who's grooming. So what's going on there? What's actually happening? And it turns out that's the social context. So these neurons are signaling, the population is signaling differently depending on whether the nearby neighbor who's watching is a male or a female. So we know that monkeys care about that. They care about who's around, and that shapes the behavior that they engage in. And we see that neurons in both of these areas, temporal cortex and prefrontal cortex, are conveying that information across the population. Again, we can decode the social context from population activity as well. So it's there, it's rich, and it is something that's accessible and potentially contributing to the actual behavior of the animal. Now, one of the things we were concerned about, of course, is whether, well, maybe what's going on here, because you're not controlling the behavior and not controlling the stimuli that are actually bombarding the animal, that maybe all of this modulation and even that information, latent information encoding across the population might reflect just the vigor of movement, for example, or what the monkey's looking at. So we used computer vision technology to track monkeys' movements as well as their field of view from their head. And to make a long story short, what we find here, for example, if you look all the way at the graph on the far right in green, that we can decode behavioral states and also social context at a very high level, even when we already regress out all of the movement patterns, all of the field of view, et cetera. And as another kind of rejoinder to that, monkeys can be looking at very different things while engaging in the same behavior, and the neuronal activity does not care about that. Okay, so it's unmodulated like that. So we think we've effectively shown that you can't completely reduce this information encoding at the individual unit level or across the population to simple sensory and motor variables. Now, one of the behaviors, of course, that we were most interested in, has never been studied neurobiologically, is grooming behavior. It's the foundation of primate social relationships, right? This is going out and getting a beer or coffee with your friend. Monkeys spend most of their time grooming each other when they're not doing other things. And that grooming is an investment. And so we looked at that very carefully. And what we found is really fascinating, because basically what these monkeys show is complete reciprocity, complete equity in their grooming. How often they groom one monkey, they'll pay that back in equivalent amounts of time. This has been observed in the wild before, but nobody's ever had the granularity and the completeness of data we have. So we studied every single grooming interaction between the animals over weeks, and they were not together outside of the experiment. And what we find is that within a session, sometimes one monkey's getting more of the grooming than he's giving. And then sometimes you'll see a flip or an inverse in that. So sometimes this equality is achieved within a session, over the course of a couple of hours, but sometimes it actually takes days or weeks, as you can see on the right. So when one monkey's down a little bit in terms of getting groomed, you'll see that repaid later. So the implication is monkeys are maintaining a ledger, a running account, if you will, of their investments and withdrawals in this social relationship. It is thought to have been kind of outside the cognitive capabilities of monkeys to be able to do this, but we had the chance to actually ask that question. And kind of to make a long story short, we use ridge regression to see if we could develop a model that could, based on neural firing rate data, actually predict who's grooming whom and for how long at any one point in time. And the long story short here is, if you look at the middle panel, we were able to basically fully replicate the patterns of giving and getting groomed in these monkeys in the short and long term, based on the firing rate data of these neurons in both populations. And for the aficionados out there, we had much better predictions of the number of grooming bouts rather than amount of time that was actually spent grooming. But we also noticed that time itself was encoded incredibly faithfully when monkeys were grooming each other, as is shown on the upper panel here on the upper right. During one session, you can see a beautiful graded succession of time during grooming, and we don't see any kind of clear, organized information encoding about time when monkeys are doing other things like resting, et cetera. And so this makes a lot of sense. This is something monkeys, their behavior suggests they track, they encode, they remember, right? And and in such a way and with in such detail and precision that they maintain a running equality in their grooming relationship. Now, we don't know if this occurs in more widely. There are a lot of other animals around, but at least when there are pairs of monkeys, it does. We looked at a few other things that are of interest to other people. It does too, but we looked at what might be involved in social support, because of course, we've demonstrated that having more social support is critical to dealing with threats, to dealing with stressors. Our kind of proxy for stressful event is a human intruder who would come in and either threaten the subject monkey or the partner. And when we do that, this is my graduate student Felipe, basically when he threatens the subject, that monkey becomes very agitated. But if he threatens his partner, the monkey also becomes very agitated. Agitation is reduced in the presence of your partner, of your friend. Okay, and that's consistent with what's been observed in monkeys and in humans in the past. And what we found, which was super interesting, is that the kind of the brain states, okay, just looking at modulation brain-wide, when in response to a threat or during a threat, the brain is very different, both of these brain areas, in terms of patterns of activation, than when you don't have a friend present, than when you do have a friend present, when basically baseline kind of signaling is maintained. So you can think of this as sort of like the brain's alarm bells are going off when you're threatened, but having a partner there reduces the sort of magnitude, if you will, of those alarms. And just to point out, I know I want to have time for questions, but to point out that at the neural level, one of the things we found that was very interesting is that the responses across the population to a threat to oneself were closest in their pattern to when the partner was threatened, okay? And they were not very similar to other kinds of behavioral states, whether that's traveling or foraging, grooming, or getting groomed. And this is a kind of mirroring, if you will, that has been kind of postulated to be really important for a variety of different aspects of social relationship. And maybe I'll put this in quotes here, maybe something like a neural correlative empathy in these animals, this mirroring between neural states and behavioral states. And again, just like before, this mirroring of neuronal activity in response to threat to self or threat to partner is independent of where the actual monkey is looking. Okay, so it does not seem to be stimulus driven, right? The threat from the actual human intruder himself. Okay, so that was the sort of what we found in these two different brain areas. One area, TEO, which is classically part of higher order visual cortex, and the ventrolateral prefrontal cortex, which is kind of been implicated in a variety of different kind of higher order social information processing. And we were a bit surprised by that, of course, as I'll talk about in a moment, that these two areas were virtually indistinguishable. And it does raise interesting questions about whether these social, behavioral, and social context states are whether the information is available kind of cortex wide, or whether that's specific to this particular circuit, because the area TEO does project to ventrolateral prefrontal cortex, which itself projects back to TEO. So you can imagine an interaction there that's shaping the activity in those two areas. And of course, our deeper interest is investigating these areas that, like the mid-superior temporal sulcus that we've studied in the past, and the anterior cingulate cortex, that have been more directly implicated in the social behavioral changes, for example, that we've observed on Cayo Santiago Island in response to the acute and long-term stress of a hurricane and degraded environment. So we were doing that. It took more technology development because these areas that are of interest for us, like the mid-superior temporal sulcus, are deep. Utah rays are only useful for recording on the surface of the brain. And so our selection of areas was somewhat, in the prior study, was somewhat limited or guided by those limitations, I should say. So we just wrapped up a study of the mid-superior temporal sulcus using depth recording arrays here from Matrix. They had a lot of problems. We managed to get pretty good data from the mid-superior temporal sulcus. That data is just undergoing processing. So we've done two things. We've now targeted more directly that social brain network, and we've also expanded the sort of social domains that these animals are experiencing by moving to an open field environment, as you can see here, schematized, that's surrounded by 52 cameras, which are used we use computer vision which works really, really well now to not only automatically quantify movement, but also behaviors and social behaviors. And we scaled this up now so that we completed studies with 3 monkeys in the arena, recording from 2 of them with a third individual, so that monkeys might be involved in an interaction or observing an interaction themselves, kind of getting closer to the reality of the field. And just as sort of a teaser, we found that, at least overall, a pattern, it looks somewhat similar to what we observed in TEO and ventrolateral prefrontal cortex, in the sense of this sort of global multiplexing of behavioral states and social contexts by the populations of these neurons. I'm going to leave it at that because we haven't finished those analyses. But those observations, as well as observations from a number of other studies that were published in the past year or so, they raise a lot of really interesting questions, both kind of socially, behaviorally, which is my primary interest, for example, the grooming, mental accounting of grooming. But for people interested in, for example, visual processing, this has been a real eye-opener, if you will, in the sense that many studies have now shown that during more natural behaviors, that neurons in a number of different parts of the visual system are actually modulated by these behavioral states and social contexts. And so that raises questions about, well, why? And that's very, we're at the realm of speculation right now. But one potential idea is that all of that information about behavioral states and social contexts might be very useful for essentially contextualizing what one is seeing, what one's perceiving, and then subsequent processing of that information, how it ends up guiding behavior, or it's being stored in memory because we know that those states and then the associated sensory experience and context are later shape subsequent behavior, even days and weeks ahead. So I'm going to go ahead and sum up so that we have some time for questions. I'm sure there will be many. Which to say that overall, well, first of all, social support is key to success. It's key to mental health and physical health and well-being. And that's true in us, and it's true in our non-human primate cousins, in particular, these rhesus macaques. And we showed that really, I think, powerfully in response to a real devastating natural disaster. In terms of what neurons in the parts of the brain that seem to be changed by or in association with changes in patterns of social behavior, that neurons, in perhaps many parts of the cortex are encoding information about dozens of natural behavioral states. It seems to be a distributed code, as I mentioned, and it's not really reducible to just movements or visual inputs. So a lot of people are concerned about the lack of controls, and I think we're kind of showing that it's not easy to say this is just something, a confound, if you will. We showed that social support dampens threat-induced agitation as well as neural activity associated with it. It looks like a mirroring type of code that may be similar or a precursor to empathy in humans. And that, for me, really remarkably, what's really exciting is this discovery of a ledger, if you will, an account that keeps a running tally of investments and withdrawals in grooming behavior, which is critical for maintaining equity, equality, and that's something that people are concerned about as well, as we know from many different types of observations. And I just want to point out that these new technological advances, both the recording technology, computer vision technology, really enables a neuroethology of primate behavior. I would be remiss if I didn't mention a few of the people who were really instrumental in doing the work. Camille Testard, who is a PhD student in my lab, is a fellow in the Harvard Society of Fellows now. She did, believe it or not, all the work on CAIO, the response to the hurricane, the survivorship, also the changes in brain or the variation in brain structure as a function of friendship was her work, but she also led the work in the lab in terms of free-moving electrophysiology. Sebastian Tremblay, postdoc extraordinaire, he's faculty up at the Laval Institute now. He developed all the technology for doing this. Felipe Perotti is our computer vision expert, has really developed a lot of those models. Kristen Gardner, veterinarian here, and important part of our research group. So with that, I'm going to go ahead and I guess I'll just stop sharing. So should I stop sharing and take questions? I guess I'll stop sharing. Okay.
Sure, sounds good. All right, thanks so much, Dr. Platt, for that excellent talk. We've received a bunch of questions from the audience, and I'll share as many as I can. As a reminder to the audience, you can submit your questions or comments using the Q&A function at the bottom of your screen. So one first question, do you have any evidence that the hurricane served as a bottleneck and therefore effectively changed the genetic makeup of the population by selecting against any genomic variants linked to atypical social behaviors?
This is a great question. So of course, we're super interested in that. And we have the data, we just have not analyzed that yet. If anybody wants to dive in, even though we have something like 90 people working on this project, there's people working on a lot of different things. So that is underway. And we now have had, it's interesting because I think it's just an ideal setup here because we have DNA on more than 1,000 monkeys. We've done whole genome sequencing. And we have all the other tissue data and on almost 600 monkeys at this point. But we had mothers who were pregnant during the hurricane, we had some who gave birth just before, actually during, and then after, and then we've had successive cohorts born afterward. The infants have not been a major focus for us, but I think you can see how we can tee up that sort of intergenerational kind of transfer of trauma, if you will, and dissociate that from the experience itself. I'll note one other thing, which has been curious, which is there's always been a very rare mutant on the island. We do have a lot of kind of monkeys in our prior work have shown mutations in genes that are implicated in autism in humans, for example. And a lot of those monkeys tend to be asocial or different in a variety of ways. So that's something that is going to be interesting to track here. But the other I was going to bring up is blonde monkeys. So we've seen, and it seems to be an increase in the frequency of light-colored monkeys, which would be potentially an adaptive response to the intense sunlight. So it's there. I'm sorry, I don't have the data for you yet. Maybe the analyses yet.
Another question. The reciprocity of grooming behavior is really interesting. The question is, did you look at this by subgroup? So for example, low status by high status. I know that study was just a few individuals, so that might not have been possible. That's related to a question I had about if you've seen or might expect individual variation in calculated reciprocity and how that might relate to kind of social success and perhaps even translation to what you might see in humans.
Yeah, this is an awesome question. So we were not capable of looking at the paper we published last year. We just had these pairs of monkeys. So that really wouldn't allow us to show that. The trios that we just studied over the last year or so in the next phase of that experiment were selected so that there is variation in their relationships. So ultimately, the idea is to get at this multi-dimensionality of social relationships. We know from prior ethological studies in the wild that status, for instance, status and maybe we'll call it allyship, grooming interact. So that if I'm a low-ranking monkey, this goes back to the work of my PhD advisor, Robert Seifert. If I'm a low-ranking monkey, my objective is to try to groom high-ranking monkeys. So you try to cultivate, favor, build a relationship, get a friend. It's like having the guy who's going to protect you from bullies in high school. And we know that works. So, but a monkey might have to invest like 10 times as much in that high-ranking monkey to get a little grooming back, but it's more likely to translate into support in a fight later on. So we'll be looking at that too.
Great. Let's see, many questions here I have to sort through. One is, do you think some of the physiological changes that we're seeing after the hurricane, so for example, with hip flexion, was due partially to ways they were engaging with their environment? So for example, less climbing in addition to stress-induced changes.
Yeah, these are all great questions. I don't know that, this is actually something I just asked my student early this morning to look at, again, to look at just whether movement overall might have changed. The environment was pretty darn disrupted, especially it took about a year with a lot of volunteers going down to remove a lot of the debris and detritus and get things cleaned up. So it might have been a bit more difficult environment. And then perhaps, yeah, it's possible that they're moving less, but it's such a drastic change. And, again, we're connecting circumstantial dots. I would love to have more causal data on that, but seeing what we see at the level of gene expression, for example, indicating an increase, or being associated with increase in inflammation. We've got other measures that suggest increase in inflammation, and then we see that this, their hip extension is getting worse. They're less flexible overall. They kind of tie together.
Great, thanks. Another question, how do you distinguish neural signals of timekeeping from behavioral heuristics like groom until reciprocated?
Well, in so what I presented in terms of that data, we were literally just looking at the progression of time during like a clock during grooming, okay? And there was very clear graded changes in activity. And when we looked at just clock time during rest, for example, or other kinds of behaviors, we did not see that. And I just want to reiterate that monkeys did not always reciprocate right away. So sometimes a monkey would be in a deficit, a grooming deficit for several days, and then would make that up later. Why? We have no idea what other things are going on. There's another, that may be conditioned on which monkey is watching them, right? So that maybe I don't want to be seen actively. I mean, this is very anthropomorphic, but don't want to be seen actively grooming that other individual in the presence of this individual. We have, for example, when we now this study with 3 monkeys in it, and then we've also done studies where we have the subject monkey watching 3 other monkeys interact. Just watching, well, I'm going to say it. They look jealous. A female who looked totally calm was very happy to be with those 2 other monkeys when they're outside and watching, completely agitated, and just is a very different experience. So there's more to it than that. It's just going to take a lot to dig into that.
Great, thanks. And maybe this will be our last question for today, again, on the topic of kind of translating this information to humans and potential kind of issues with social cognition. So kind of what you see as potential paths for translation there, especially for kind of loneliness and stressful and kind of trauma-related situations.
Yeah, I mean, I think there's several different ways in which this can have translational relevance. I mean, for example, looking at particular genotype phenotype combinations that are associated with differences or impairments in social competence, for example, as you see in autism or other disorders, schizophrenia, et cetera. I think that we can begin to do some modeling of therapeutics. In parallel work, we've developed and have pretty high fidelity, behaviorally relevant control of these circuits using optogenetics now, monkeys, and also using dreads to manipulate these circuits in a cell type specific ways. So I think it's sort of a clinically and medically kind of relevant translation. That's one direction. The other, in terms of loneliness and trauma, yeah, I could say things about, oh yeah, we can develop small molecule pill or whatever. I don't think that's like where we're heading. To me, it's there are many things that jump out of this. One is that this stuff is deeply baked into our biology. It's not just some function of our culture or our politics or anything like that, right? That's why I think the impacts are so inescapable. And to me, the remedies that are that seem the most powerful are sort of behavioral and social, right? So we know that from other human data that's been coming out. So increasing equality, for example, reduces stress and reduces even the emergence of psychiatric disorders. The same thing for access to greenery, to having a clean, green, healthy environment to live in. So we put all of that's where I tend to put my emphasis, and that's my job at Wharton, is to make this case not only to policymakers, but to business leaders, and we're doing that, to make those investments in the private sector to build essentially brain capital for the future.
Great. Thank you so much, Dr. Platt. And now I'll turn it over to Dr. Simoni for closing remarks.
Thank you, Dr. Bertha-Horowitz. That was great facilitating of excellent questions. We didn't get to them all. I hope they can reach out directly to Dr. Platt if they would like to further pursue their questions. But Dr. Platt, thank you very much. That was an excellent presentation. Just like to remind everyone that a copy of this presentation will be available on OBSSR's website in about a month or so. And otherwise, that concludes our webinar today. Thank you very much for joining, everybody.
Thank you.
Take care.